Okay, so today for the series it was StatPhys in Florence next July. We interviewed Daniel Maria Busiello, who is a group leader in the Department of Physics and Astronomy, University of Padua, previously in Max Planck Institute in Dresden. And so, Daniel, can you please introduce yourself, your research, your career, your interests?
So thanks for this opportunity and invitation to participate to this interview. So I studied in Padua during my PhD, mostly on stochastic thermodynamics. And then I moved to Switzerland, to EPFL, starting to investigate the idea of applying this method of stochastic thermodynamics to quantify, study, and understand the role of dissipation and non-equilibrium phenomena in chemical systems. And then I moved to Max Planck, as you mentioned, where I combined these ideas of studying chemical systems and biochemical processing to the idea of that biological systems and chemical systems convert inputs into certain output or certain responses. So also facing the idea of studying information theory in this kind of systems. And now more or less my research span this area, so stochastic thermodynamics, information theory, and chemical systems, trying to combine these ideas into the formulation of a characterization of processing of external stimulations in biochemical networks.
So you are applying ideas of non-equilibrium statistical mechanics, statistical physics to chemical systems, which are traditionally rather related to equilibrium. So can you explain the importance of non-equilibrium in chemical systems?
So my studies on this topic started with the idea of the original life problem, actually, and also the question how life has originated at its dawn. So this question, we try to answer this question by investigating the idea and exploring the idea that an energy dissipation um, started and sparked favored the certain processes that lead to the formation of complex systems. So if you imagine that the equilibrium, if you have a complex systems in general, this tends to relax and form its microscopic components with a lower energy. Out of equilibrium, like injecting energy in the system, you can actually push all these building blocks into the formation of a stable, complex system that will something that is closer to what we observe nowadays.
See, actually, life is a kind of non-equilibrium pattern, no? In some sense.
Exactly. So this was the starting of our ideas, and then we started playing around with very simple models to understand if this actually was a possibility. So if it's possible to select some complex state in some general sense using non-equilibrium phenomena, non-equilibrium mechanism. So this was the starting idea. Then we also applied this to an experimental systems, a very simple ones, where we basically studied the stability in non-equilibrium conditions of the Faranos, which is something which is observed in nature, and we actually found that this can be stabilized through non-equilibrium mechanisms. So this is actually possible. So this idea that we had in the beginning. I also applied all these tools of non-equilibrium in more artificial chemical systems, if you want. So more related to the idea of building batteries that operates with pH differences, also in collaboration with some experimentalists. And I actually realized that using tools on non-equilibrium physics to face problems and to study systems that are already analyzed experimentally provide a very powerful tool to understand what's happening. They can really override the standard ideas of using pseudo equilibrium quantities or pseudo
Mhm.
equilibrium chemistry to understand these systems. So it is true that from a physics perspective, it might be obvious that the system operates out of equilibrium, but to have a proper chemical modeling and proper computation of all these quantities and how to one can use non-equilibrium or identify non-equilibrium directly from experiments or from data it's actually a non-trivial task and can give really powerful insights on how a system is operating, and also to give predictions on what we should expect in other experiments.
So you mentioned information theory, which is also in some sense related to the recent discoveries now on how we can predict shapes and reactions in chemistry, biology, and so on. And I think that what you study are related clearly also to cells, neurons, and many other biological systems. Can you illustrate what you do on these topics?
So basically, just following my trajectory, basically, I started investigating information theory from signaling networks. So biochemical network that takes some input, convert it into an output by using, as I said, like as I mentioned before, all these non-equilibrium mechanisms. So this was the connection point between non-equilibrium in chemistry and the information theoretic part. But then after that, like starting with the ideas of exploring adaptation in chemical systems or habituation phenomena, which is the decrease of a certain response when you have a repeated stimulation of the system. We basically realized that information processing is everywhere, not just in biochemical systems, and a theory that tackles the information processing might actually be more general than just studying biochemistry or chemical signals. So that's why I moved a little bit the exploration towards the idea of studying information processing and information or using information theoretic tools to general frameworks of stochastic processes. And then this can, of course, encompass the modeling of biological systems or the modeling of more artificial systems, closer to the idea of implementing neural networks that make decisions, or towards this line of research. So my background is mostly on the biological and biochemical side, but now the idea of my research in this direction is always, I mean, it's more and more towards the identification of key ingredients that underlie the propagation information or they underlie the processing performances in general systems without adding the specificity of some of the like specific example, the detailed examples that I'm
A
going to
more
study.
general theory in some sense.
Yeah, exactly. Because in the end, all these information processing systems, they share a lot of similarities, at least in how they work and what should they do in principle. So there should be some mechanisms that are common to all of them, that are actually important and crucial to transmit and propagate information in an efficient way. And these mechanisms are related to the presence of multiple timescales, to the presence of a certain non-equilibrium processes that happen inside the systems, sometimes also to the presence of dissipation when we are able to properly identify and quantify dissipation in the systems. So there are multiple ingredients that are basically everywhere. So this means that they should play a proper role in shaping these systems towards a biological functioning that we see and it's emerging.
Okay, so finally, can you illustrate in a few words what are you bringing to next StatPhys?
I'm bringing a work on this topic. So on the idea that you have an input arriving at the systems and you want to understand how this input is converted. And this work explores the role of nonlinear operations inside the system. So many models of biological systems and also non-biological ones, from neural networks to gene networks to ecological networks, all of them share nonlinear features and nonlinear couplings inside the system. But exploring the role of these nonlinear operations is actually not easy to do analytically. And even numerically, when the system is very high dimensional, it's a challenging task. So the work that I'm going to present in StatPhys is a first step in this direction, trying to understand the differences between a system performing integration of external inputs and a system performing the simultaneous parallel computation of different signals. So these two are two different schemes that are both implemented in biological systems. And so understanding the performances and the difference between the two might shed light on why a system is implementing one method or the other method, since both of them are actually found in biology, and both of them are actually used to model systems when sometimes the microscopic determinants are not known or accessible.
Okay, so thank you. I'm really eager to hear you here in Florence next July, and so I hope we shall meet and discuss.
Sure. Thanks a lot, and see you in Florence.
Okay.